Good afternoon to all my students. Today I was going to deliver one of the most interesting topic, that is the modes of DNA replication. That is modes of DNA replication. So the modes of DNA replication, also the called the types of the DNA replication. So as we know that in 1953, J.D. Watson, an American biologist, and F.H.C. Crick, the British physiologist, proposed the three dimension of the model of the physiological DNA. Three dimension of the model. That means the modes of the DNA replication. So in the Watson and Crick in 1953, there is a three models is there. One is first is, that is, as we know that DNA is a double stranded. DNA is a double strand. Okay. So this is the sense and the nonsense. Okay, by antisense. This is called antisense DNA strand. The 5 prime to 3 prime, and 3 prime to 5 prime. These are the DNA double strand, and it is replicate. So replicate is the one part. That is the one part of the cell cycle. Okay. So this is the DNA replication. The modes of the DNA replication. There is the three types of the DNA replication is there. One is semi conservative DNA replication. So semi-conservative DNA replication is the first modes of the DNA replication. That means is a semi, and conservative. That means these are preserve or conserve. So this is two strand is there. Two strand DNA is two strand is there, and these two strand they are replicate. Okay. See these two strand, they are separated. These are one strand, and this is the two strand. This is the one, and this is two. They are this in the hydrogen bond is there. Okay. So hydrogen bond is there. So this is the first strand. This is the second strand. Okay. And the new strand is formed. This is also, there is another new strand is formed. This new strand already formed is there. So it is the black color strand. It is the black color strand is here. That is the first strand, and there is the second black color strand is here. This is the new strand, new synthesized strand is formed. This is the red in color. This is new strand. So it's called first strand, black color strand is called the parental strand, and the red color strand is called newly synthesized strand. Okay, this is also the black color is the parental strands, and the red color strand is the newly synthesized strand. Okay. So as you know that there already one strand is newly formed. Here is also one strand is newly formed, and in this two strand, DNA of this two strand, one strand is conserved here, this black color strand, and here also new strand is synthesized. So half of the strand already separated to each other. There is the one, here is the one, half of the strand. So this synthesis, this conservative, this replication is called semi. Semi means that is the one strand here and one strand here. That is the half one strand is here, one strand here. So this is called semi conservative. Conservative DNA replication. Semi-conservative DNA replication. This is the semi. Why it is called semi? Because here is the one strand is here and one strand is here, and newly strand already formed. This replication is formed the semi-conservative replication. This replication is proposed by Watson and Crick. This is the scientist. These are from this type of the semi-conservative replication. This is the first the modes of the DNA replication. They propose this type of the replication by Watson and Crick. And that is the second DNA replication model is called, that is conservative. That means conservative DNA replication. So this is the double stranded DNA. So this is the double stranded DNA. This is the 5' to 3' to 5'. Okay. This is also, there is a hydrogen bond is there, hydrogen bond. Okay. And by this process, this is the conservative process. By this process is here the conservative. That means there this model already proposed by Max Delbert. The semi Max Delbert. These are Max Delbert. This is the proposed by the third group of the scientists. This is the mainly proposed by this type of this model. That means there two strand already copied here. Two strand here, just write. Two strand are copied here. Two strand are copied. Here is the first and the second. This is also first and second. They are not separated. They are same. That is the same in condition. This is the first. And here newly synthesized two strand are formed. This is the red color. Newly synthesized two strand are formed. So you just look here. This is conservative. That means total DNA, the two strand total DNA are copied here, and here that is the newly two strand are formed. So this is called conservative DNA replication. Conservative DNA replication. That means they are not unwinding, they are not separated, these two strand. They are copied to here, the two strand in here, and here already newly two strand are formed. They are separated in this two strand. So this type of replication is called conservative replication. These are conservative replication. Okay, this is conservative. They are not separated. Okay. So conservative replication, that means newly two strand are synthesized separated. They are not attached with the parental strand. This is the parental strand. Both strand are parental strand. Parental strand. And this is the newly synthesized strands. Okay. This type of the modes of DNA replication is called, that is the conservative DNA replication. And number third, that is the तीन नंबर जो replication है वो है disperse. That means the disperse replication. So this is called dispersive. This is called dispersive. This is called dispersive replication. Dispersive replication. That is dispersive. That is dispersive DNA replication. replication. Okay, so in this replication we already said that it is proposed by Max Delbach as the this type of this semi-conservative model will generate a mass where unwounded the DNA black bond is the broken, the stand one copy into the short segment two nucleotides that is generating, and the network with the needs of the separated. That's mean here two stand is there, two stand, two stand. Okay. This is the two stand of the DNA. It's the 5' to 3', 3', 5'. Okay. This is the two stand. Okay. These are dispersed. That means they are scattered. That means they are scattered form. That means they are scattered form. Okay. That means they are scattered. Okay. That means they are scattered. That means they are scattered form. This is the newly synthesized DNA part, and this is the parental DNA part. That is the old DNA part. This is also called the old DNA part. This is the old DNA part. This portion is the black color. This portion also the black color. These are old DNA part, and this also the black color old DNA part. And this is the red color. This is the new synthesized. This red color new synthesized. That means they are scattered. There is the red, then black, then red, then black, the red. They are scattered. Okay. One stand, they are not in totally red in color, totally black in color, but they have some portion already black color, some portion already red color. That means they are scattered in form. And here also there are two stand. They are also replicate. They are red, and here also the red, and this is the black, and this is black, this is the black, so this is the red, and this is the black. Okay, these are also newly synthesized stand, and this is also the old stand. But here there is the three modes of the DNA replication. That means there are the three different scientists. They are showing these three types of the replication. One is the semi-conservative, and another is the conservative, and number three is the dispersive DNA replication. But this is the most acceptable and the proof leaf. This replication, DNA replication, this is the semi-conservative replication. After some experiment, we proved that the DNA replication is happened only the one process. This process is called the semi-conservative DNA replication. There is the another two process, that is dispersive process and also the conservative process. These process are not the proof by this experiment. So we are all that proved this experiment that is DNA replication only happened in semi-conservative modes. Okay. In my next slide, we are discussing about this test of the Michelson, the lab test, where the DNA, where the proof that the DNA replication is the semi-conservative replication. Thank you, all of you.